pick up where we left off. I didn't want to run out of time in the last video talking about the second law. YouTube only has a 15-minute limit, so I didn't want to take 10 or 15 seconds in case we needed a little extra time. So here we go, law of dominance, and this one's probably the easiest one to get, but I just wanted to make sure that I had time to explain it fully. The law of dominance is pretty simple. It is basically talking about the hybrid organisms. In other words, an example, big T, little t, like those offspring in a couple of slides ago. They are expressing the dominant allele. For example, they look tall. Okay, they're looking like the dominant form, right? That is the law of dominance. Again, pretty simple, but something that could be easily mistaken. If you see a big T and a little t, you would think that they're not tall or not short. They're like in between, right? But it's not the way it is for these types of genes. Law of dominance states that the hybrid organisms, big A, big B, little b, doesn't matter what the letters are, but if they're hybrid, or now you know heterozygous, they're going to express the dominant allele. Okay, important to recognize that this law, what it means and what it's talking about, not just for this little example here, but we're going to refer to it basically every single time we talk about genetics. 1905, again, not really a date you need to know, but it kind of emphasizes the idea that it took a while for genetics to kind of pick up in the scientific world. It took a while for genetics to kind of overcome the kind of the hangover from the evolution hype that happened during the 1800s. Okay, so this guy, again, not that you need to know his name, but that's where the name comes from, Punnett squares, I'm hoping you've seen before, came up with an easy way to determine genotype proportions. Right, important for genetics to figure out, you know, what are these results going to be. So here's a pretty easy example. We take a heterozygous parents, big T, little t, they're both the same, cross them. So that simply means that we are going to make the gametes. Alright, one allele from one chromosome goes to the left, the other allele, again, from the other chromosome, or sister chromatid, I guess, goes to the right. Same thing for the other, but top and bottom. Alright, and then simply just matching them up. Two big T's here in the first box, big T and little t in the second box. Down here the first box is big T, little t. Something to recognize here, it is not little t, big T. Okay, you always want to write the capital letter first, otherwise it's going to start to get really confusing, especially when we get to more than one trait. And then the last box, little t, little t. Alright, so important to recognize that you'll always have capital letter before lowercase letter in the Punnett square. And again, when you're going through this quickly and you have one is big T, little t, the other is little t, big T, at first glance they might look different when really they're not. So here's what this stands for, and this is probably something you want to write down underneath here. G stands for the genotype ratio. Okay, genotype right here, the genotype ratio. You get 50% big T, little t, that's these guys, 25% big T, and 25% little t, little t. Or in other words, this would be a 2 to 1 to 1 ratio. Phenotype ratio, which is capital P here, again, probably going to write this down, you're going to see this a bunch, P equals 3 tall to 1 short, a 3 to 1 ratio. Right, that's something you've already seen in the previous slide, a 3 to 1 ratio when you cross hybrids. 3 tall, that's right here, law of dominance, right? All these guys are going to look tall because they have at least one capital T, the other T doesn't matter as far as their phenotype. And then 1 short. So, we're going to get off of these 1 trait crosses, those are the easy ones. We're going to get into 2 trait crosses. Monohybrid crosses are easy, separating alleles according to Mendel's law of segregation is a piece of cake. If you have a heterozygous parent, like you just saw in the previous slide, big T, little t times big T, little t, you just separate big T from little t. Okay, that's half and half. Pretty simple, you just split it in half. But for 2 traits, it's a little tricky, especially dihybrid, meaning one parent has 4 letters. Hybrid for one trait and hybrid for another trait. The gamete separation gets a little bit harder. Actually, it gets a lot harder. So, let's have an example. 2 parents that are heterozygous for seed shape. We're just going to use a capital R and lowercase r. Again, you want to use letters that are just different from each other. Using Y's may not be good if depending on your writing. Using C's are bad. Letters O would be bad. Letters like that, Z and stuff, that looks similar if you're writing too fast. So letter R we're going to use for seed shape. Letter Y for seed color. So their whole genotype has to have seed shape and seed color in it. Is big R, little r, big Y, little y. Again, dihybrid. Hybrid for two traits. Seed shape and seed color. Now we're tracking two traits from one generation to the next. It just got a lot harder. So we have to separate the alleles. Again, you can't just split them in half. If you split them in half, you're giving only seed shape to one half and only seed color to the other half. Doesn't make any sense. So we're using the FOIL method from algebra. First, outer, inner, last. If you forgot what that means, you probably want to write that down in your notes. So here are our parents. They have the same genotype, so we can assume that they will make the same gametes, in the same ratio at least. So the 4 different gametes we will get using the FOIL method. Here we go. The first refers to the first 2 letters that are different. Again, half of the genetic information we want to pass on seed shape information, 1R, and seed color information, 1Y. So all of our gametes, all of our sex cells have to have half the genetic information, but that means half seed shape information, half seed color information. So each gamete produces an R and a Y. So the first will be big R, big Y. Here's our firsts. Now you might want to off to the side make this little diagram, or make this genotype, but then put arrows. Firsts are these guys, big R, big Y. Okay, there's our firsts. What are our outers going to be? Outer meaning the very outermost letters, right? So big R and little y. What do you think the inners are going to be? Hopefully you're catching on. It's going to be little r, big Y, and then process of elimination, but also the lasts of each, right? The last R and the last Y. And again, what you have here, all different combinations of seed shape and seed color being passed on to the offspring. And now we have 4, because our parents, now we're talking about tracking two different traits that each have 2 different alleles. So you have to have 4 combinations. And here are basically those phenotypes. Okay, round and yellow, round and green, so same seed shape, different color, and the opposite, wrinkled in yellow and wrinkled in green. So again, we have 4 different phenotypes and 4 different gametes. So things just got a little bit harder. So first to go through kind of what happened when Mendel crossed these. Again, he crossed round wrinkled in yellow and green, did exactly what you just saw. Genotype of the purebred or homozygous, what is it? If you're looking at round and yellow, I capitalized them for your hint. Round and yellow genotype is for purebred, or otherwise known as homozygous. It's going to have 2 R's and 2 yellows, or sorry, 2 Y's. So big R, and big Y. Again, purebred for both, homozygous for both. So the opposite, genotype of a purebred plant with wrinkled green seeds. Again, these are both the recessive forms. So 2 little R's, 2 little Y's. Again, you have to have 2 letters for each. And now here's where it gets interesting. When you cross these 2 genotypes, or by phenotype, round yellow with wrinkled green, you get a very interesting, unique, and something you're going to want to remember ratio. All of the F1 offspring were the result of this phenotype, 100% round and yellow, but the genotype is where you see a big difference. Okay, crossing the F1s, crossing all these guys round and yellow, now we see a big difference in genotype. If you cross all round and yellows from these parents, they're all round and yellow, because they have to have big R from one parent and big Y from this parent, so they're all the offspring are round and yellow. But now crossing these guys, and again these are dihybrid, big R, big Y, little y, gives us a very unique ratio of 9 to 3 to 3 to 1 in the phenotype. We'll see that in the Punnett square in just a second. Gives us basically Mendel's last law. This is a typo on my slide, it's good on yours. This is the law of independent assortment, and you've already seen this in meiosis. The law of independent, here's how you spell that, independent assortment. And again, we've already seen this in meiosis during metaphase 1 and metaphase 2, when these chromosomes line up. But again, Mendel didn't know this stuff. He didn't know about chromosomes, so his law of independent assortment states that genes for different traits are inherited independently of each other. Let me show you what that looks like in a Punnett square or diagram first. Okay, here's what I was talking about, those round yellows. Parents times wrinkled green parents. Again, purebred dominant, purebred recessive, times each other. You get all the offspring looking like the dominant form. Again, this is law of dominance. These guys are big R, little y, little y. Both of these. Okay, big R, little y. Cross these together, and this is where the mix-up or the strange but unique scenario takes place. In the F2s, you get 9 that are going to be round in yellow because they're getting at least a big R and a big Y. The other letters don't matter. That's what the underline stands for in all these. 3 that are round in green. Doesn't matter what that second R is. Could be big or little. Still going to get round in green. Same for wrinkled in yellow. And then the runt of the litter is going to be the wrinkled in green. It has to get 2 little R's. Has to get 2 little Y's. Again, law of dominance to be the recessive form, wrinkled in green. So, here's our situation. Same exact scenario. Here's the parents. Big R, little y. Here's those F1s that you just saw. Big R, little y, little y. So what are the gametes going to look like? Again, FOIL method, right? So firsts are going to be big R, big Y. And then your outers, big R, little y. And since the parents are the same, you can just do these the same for each. First box, second box, or first row, first column. They're going to be the same. And the third ones, where are we at? The last, or no, inners, sorry. Inner, little r, big Y. Now the last, little r, little y. Now you see why this guy got the wrinkled green phenotype. So, the genotypes are just going to pop up on their own, and just about every second or so. See if you can figure out what each genotype is going to be. I'll give you the first one. Again, you got to put R's together, so this one's going to be big R, big Y, big Y. What's the next one going to look like? Well, we're going to go left to right as we go down. Here we go. And what you'll notice for phenotypes is you start to get a triangle pattern. And it's only if you set up the gametes correctly. Again, gametes, sex cells. So literally, like, turn one of these into sperm. Or top row, turn them all into sperm. That's from the male parts. Okay, these guys are eggs. Alright, so literally, if you set these up correctly, where you foil in order correctly, to check and see that you got it right, you're going to see this triangle pattern. I'll let these genotypes finish up. But you'll see a triangle pattern with the phenotypes. Guys, genotype ratio, you'll never be asked this. You're not going to be asked what's the genotype ratio of all these, because there's way too many of them. Okay, we're just focusing on phenotype. What do they look like? Alright, and you can see this triangle pattern. Here's the 9 that are going to be round and yellow. Again, check out the letters, why they exist that way. Alright, another triangle right here. It skips the in-between, but still a triangle. That are going to be round and green. Again, they're only green if they have 2 little y's. That is the recessive. The wrinkled in yellow. Again, wrinkled means it has to have 2 little r's. Law of dominance. Again, triangle pattern. And then the last one, the runt, is 2 little r's, 2 little y's. Thanks a lot, mom and dad. I'm a freak. But, guys, notice the triangle pattern, right? If you foil correctly, then the rest of this is easy. Filling in the blanks. But again, here's the trick, putting 2 R's and then 2 Y's. The parents were 2 R's, 2 Y's, so all of the offspring have to be 2 R's, 2 Y's, and you want to do them in that order. If you start doing RY, RY for some and not for others, it gets really confusing. Okay, it's really hard to find the right genotype and match them up. So, do RRYY just like the parents. Doesn't matter if they're capital or lowercase, as you can see as we get down here. Okay, you always want to do RY order, or letter order, genotype order.